This is the Arthropods 1 vodcast on Chelserata. Did you know it takes almost 13 horseshoe crabs to get a gallon of horseshoe crab blood? Each gallon goes for about $60,000, making it one of the most expensive medical products we have. When horseshoe crabs reproduce, they gather in large groups on beaches where they are harvested.
Lumulus polyphemus, aka horseshoe crabs, are part of the order Chelicerata within phylum Arthropoda, meaning they have chelicerae. Chelicerae are appendages they use to crush food and put it in their mouths. These chelicerae are vital to their ability to eat since they are omnivores and have no teeth or mandibles to grind their food. It takes around 10 years for a horseshoe crab to reach sexual maturity. This long period where horseshoe crabs are unable to mate, combined with habitat loss, greatly impacts their population's ability to recover when harvested, which is why they are listed as vulnerable internationally. Once they are sexually mature, horseshoe crabs have a spawning season that peaks around May and June. This is when they are found on the coastlines and are able to be harvested. To harvest the horseshoe crabs, they are drained of their blood to collect the Limulus amoebocyte lysate, or LAL for short, contained within it. After the blood is harvested, the LAL forms into white pellets at the bottom of the blood collection bottles. LAL is extremely important to the medical field because it is used to test for endotoxins, which are an indicator of bacterial presence. When in the presence of endotoxins, LAL coagulates and turns the substance being tested into gel. LAL is so sensitive that it can detect and coagulate even at concentrations of parts per trillion. The vulnerable status of the horseshoe crab population and the great need for their blood is why their blood is so expensive.
For that very reason, we harvest around 500,000 horseshoe crabs a year. A study was done that looked at the effect of the bleeding process on the behavior and hemocyanin levels of horseshoe crabs. 3 stressors were tested, these being exposure to air, exposure to heat, and blood loss. During the regular transport and harvesting process, horseshoe crabs are left out of the water for significant periods of time. This is often in direct sunlight or places without temperature control, exposing them to warmer temperatures and air than they are used to. 48 male and 63 female American horseshoe crabs were harvested from June to October of 2016 and 2017. They had initial blood samples taken, and the animals were fitted with accelerometers that track their activity levels. The crabs are placed in a circular mesh enclosures filled with water and exposed to a natural light cycle. Baseline behavioral data was collected for a week before the treatment. There were multiple treatment groups consisting of combinations of the 3 stressors and a control group. The experiment went on for 2 weeks for each group tested, and each week they had hemolymph samples taken. The bleeding process consisted of a 52-hour period imitating what happens when companies bleed horseshoe crabs. For the heat exposure, the crabs are placed in barrels that were next to space heaters or direct sunlight and driven in warm trucks for 4 hours each. For air exposure, crabs are left in barrels for 16 hours at a time. Crabs exposed to air were also either covered in moist burlap or left uncovered. There were variations in baseline data and differences in temperatures, so percentage of baseline hemocyanin was used, as well as multivariate analysis of variance for effects of time and treatment. The acceleration threshold was used to determine if a crab was active or inactive. There was a mortality rate of 17% across all groups, not including the control. The highest mortality rate was in the group including all 3 stressors. It also had the largest drop in hemocyanin levels. Almost all deaths occurred 1 to 2 days post-treatment. There was an average hemocyanin level for all crabs of 40 milligrams per milliliter, and every group ended up having a decrease from the base concentration The threshold of the healthy hemocyanin levels was 13 milligrams per milliliter, and any lower led to a much lower survival probability. Those exposed to air and the control had the smallest decrease in hemocyanin levels. The relationship between hemocyanin, mortality, and activity was that lower levels led to higher mortality rates and less activity. Air and heat on their own were not found to be lethal, and the bleeding process was found to have the biggest increase in mortality rates, and heat had the most effect on dropping hemocyanin levels. Hemocyanin levels are affected by stress, so even just keeping the animals in captivity has negative effects. Hemocyanin is 90% of total hemolymph protein, and 30% of hemolymph is extracted from horseshoe crabs during the bleeding process. Though the blood is rapidly replaced, hemocyanin is not, which is why the bleeding process can be so harmful to horseshoe crabs.
The horseshoe crab has a big significance in the medical world. The Limulus amoebocyte lysate, or LAL, found within the blood is extremely valuable for medical testing. It is used for testing endotoxins in vaccines, medical devices, and other products in the scientific and medical communities. To make sure everything is as safe as possible, the horseshoe crabs are necessary. There has not been another alternative found or synthesized that has been able to replicate the abilities of LAL compound found in the crab's blood. With the decreasing horseshoe crab populations and no new alternatives found, our healthcare industry would be hugely affected, as we would not be able to prevent endotoxin-related illnesses. Not only are horseshoe crabs important to the medical and healthcare industries, but to the ecosystems they live in as well. If their population continues to decline, it would be detrimental to the health of the coastal ecosystems because their burrowing oxygenates the sediments and removes excess nutrients that cause algae blooms or other issues. Their eggs are also an important food source for shorebirds and sea turtles that depend on them, which could also impact their populations as well. They also have several symbiotic relationships with flatworms and snails. Horseshoe crabs are important to the medical industry and their ecosystems in which they live. Without them, the world would not function as it currently does, so we need to take care of them and watch how the bleeding process affects them.